All right, guys. So I'm going to go ahead and get started with the introductory PowerPoint presentation. And before we start, I do wanted to remind you guys on a couple of things as usual. Um, Next week is Gymnosperms, April 8th, and that would be your last actual lab. Um, and week after that, April 15th, is your second lab practical on plants. And that's going to happen in the same way as we did the animal practical. Um, so that will be open book. And I strongly encourage that that you use your well-prepared lab notebook for that purpose. Because like when you have multiple material, it's harder to go through all of them in an open book exam. So study early, right? And prepare early. Make sure that within the next 2 weeks that you have your lab notebook properly prepared. And do recall that your lab notebook is also due um, on the same day of the exam by the end of the day, right? And then the links to upload that is also going to be available through Blackboard, the same way you uploaded your the first half of the lab notebook. You will work on the second half of the lab notebook in the same way. You can use the same methods you employed. Some of them actually were very successful using OneNote. That's great. Some of you send me the scanned ones. That's also great. But remember, guys, when you're sending me the scanned images, put them together as a PDF, right? Separately uploading them, that's actually going to take a lot of time at your end, a lot of efforts, and it's hard for me to manually go through all of them. So do merge them together, right? I have in the syllabus, if you look up, I provide several free tools where you can actually merge these things together. So merge individual photographs together, right? So make sure, like most of you did it correctly, but a couple of you uploaded separate images, right? That's harder to grade. That's going to take too much time for both of us. So merge individual photograph if you're doing it that way. And so that's one thing, right? Again, just to revise, next week is your last lab. Week after that is in 2 weeks. On April 15th is your lab practical, plant practical. We will cover all the plant material for that one, right? And you will not be tested again on your animal material. It will be the same number of exam questions. Going to be 70 questions, graded out of 50, with 2 bonus points, optional bonus points. And that the exam will be available on the 15th, and it'll be available, few minutes before, like about 15 to 20 minutes, 15 to 30 minutes before the lab starts, and it will close about 15 to 20 minutes after the lab ends. So we are going to, so that will avoid any conflict. So you will be taking the exam during the lab time frame. Everything else is going to be in the same way, right? You did the animal practical. You will have the question set on the Blackboard exam version, and then you will have a separate PDF that includes all the photographs, right? So you, for each question, read the question, look at the photograph on the PDF, and then answer the question. So that's how it is going to go. You will also have 2 attempts in case you go through some unfortunate circumstances where you get kicked out or something. Right now, make sure when you are finishing that that you properly upload it, or properly you hit upload when you are done with the exam. And the last week of the all the labs is going to be April 29th. Speaking of which, the week after lab practical is April 19th, right? And that week we will not meet, right? April 19th, there are nothing scheduled. April 29th is actually your poster presentations, right? Now, for that one, all of you are supposed to be present, right? You might not be presenting, like there will be a presenting order, right? And I will actually let I will not pick who's going to go first. I'm going to let you decide who's going to go first, and it's based on your choice. And if you have nobody's choice, making a choice, I will actually pick teams. We only have 3 teams. So and each team will have about 15 to 20 minutes for the presentation. And when you're presenting, make sure that every member of the team is presenting. Make sure that you know what part in your poster you are presenting, and you will be sharing your poster screen by screen sharing through Zoom. So, and if you have any other prop-ups, remember, you are welcome to add other prop-ups, but when you are using those prop-ups, you may want to switch your screens from the poster to the other thing you want to show. So you have plenty of time to prepare for the poster, right? And so if you have any questions on the poster, today and next week are the best times to ask, because after that we will not meet virtually before the actual presentation date, right? So because on the lab practical date, we will not be meeting in person, right? You will be doing that on your own time. So do make sure that you have all your questions resolved regarding the poster. Make sure that you are prepared. Make sure what part you are presenting. Make sure who is presenting in which order, right? All of that is up to you to decide, right? And then on the day of the presentation, I'm going to let whoever goes first will go first. If that's how it is going to go. And also on the day of presentation, right? Yeah, you will be doing 2 things, right? If you look at the blackboard, let me actually show the blackboard to you first over here. Go to blackboard over here. Give me a shout out, guys. Are you guys looking at my blackboard screen?
Yes, I can see
Very
it.
good.
Yeah.
Yeah, great. Now in the course content, if you go to your blackboard page, let's go to, I need to make sure that I know where I'm looking at. Yeah, so we are inside the course content. If you go to research poster information, you will find these 2 Word documents at the bottom, right? Actually, let me bring it up on the top so that you can easily find it, right? Over there, you actually have 2 different forms, right? So each one of you will fill those forms. The first form is peer review sheet form. So that sheet is to review the contributions of your team members to make the poster. Right now, that content that review you do will be held in confidence. That will be not visible to your team members, right? Only I will be the only one who's actually reading that, right? And the second one, poster review assignment form, is actually let's look at that really quickly so that we know what it is. Wait until this opens up, right? So the second form that you're looking at right now over here is actually when you are reviewing other posters, right? We have 3 posters, right? Or 3 or 4 posters. I believe it's 3 posters. And so each team, each student will grade, quote unquote, grade other posters, right? Based on these questions. Those are very easy, right? It's actually not that hard to go through. You will take a maximum of 5 to 10 minutes to fill this one up, right? This is again reviewing or assessing other posters, not your own, right? You are not assessing your own posters. What you're assessing is actually over here. Let's look at this particular form. The first link, let's take a look at that. This is actually assessing not your own poster, but the contributions made by other students. Now, what am I going to do with this? These are going to, so this particular one, right? Peer review assignment form, that this is, I'm going to use this, the assessment you make. You are not grading, right? This is not you providing a grade for your peers, right? You are telling me how well did they contribute. I will take your assessment, your peer assessment, when I'm finalizing your project grade, right? That's how it is going to happen. I am the only one who's grading your project grade, right? So whatever you feedback you provide over here, I will weigh that in significantly when I determine the final student grade, right? It is not like averaging, right? It's not averaging what I give and what each of the team members give, right? It's not like that. It's actually something I do. I weigh in by considering your verbal comments, your written comments, and your actual grade points you provide over here. So I take it very seriously, right? And then when it comes to this one, and over here again, when you grade other one, right? Am I considering this? Am I averaging my grade and like every other peer post assessment? No, I am the only one who's actually assessing what your final poster grade is, right? This is simply to get an idea how well you criticize, how well you assess other people's posters, and not to mention both of those will be part of your grade, right? Like doing the peer assessment and doing the peer team assessment and doing the peer post assessment are part of your um, final lab grade, right? So where you upload it, let's look at that. If you go back to course contents, go to this particular one, right? Submit all homework and assignments, right? This is the peer assessment upload, right? This is where you assessed your members of your team, right? And this is where you upload the other two posters, other 2 or 3 posters, that is not yours, right? So because of that, doing that is extremely important. That is a part of your grade. And how you waive your team as your other team members, I will seriously weigh that in when I'm finalizing your final grade. And I will again, when I do that, am I going to mention this particular student poorly assessed you? No, I'm not going to do that. I'm going to mention, this is the overall grade I gave you, and this is the grade after considering the peer assessment. I will not reveal individual student identities, because it will held very strictly confident. Generally, I have never found a big problem in this regard. Students do contribute equally. And if you have a problem with someone not contributing, you can actually mention that, right? That's actually why the peer assessment is there, right? I mean, if you are, if there's one member who's not doing their assigned tasks, if they are not meeting the deadlines, that's a serious problem. We need to actually grade better on students who are taking up a lot of work, and have to penalize people who are not doing their assigned tasks. That's why the peer assessment is there. Any questions regarding the practical or the poster component? I'll pause for 1 second before starting back. Okay, I don't hear any questions, so we will continue on to
Wait, when is
today's.
it? When is the assignment due?
Oh, the peer assessments?
The whole thing.
Yeah, so the both peers, the peer assessment and the poster assessment, both of them you should submit on the day of the presentation. So in the links that it's set to April 15th. So April 15th, 11:59 is actually when they are not April 15th, my bad. April 29th, right? April 29th is actually when they are due. Were you going to, yeah? Any other questions? Anyone else? Any follow-up questions on that? So just my own follow-up instruction for the peer assessment is that your team assessment, right? Team assessment, you can do it before the day of the presentation, right? When everything is done, when you upload, when you are ready for the presentation, even before the presentation starts, you can actually do the peer assessment, right? You probably won't know how to do it by now, right? So you can do that part early. So because of, and that way, when on the day of the presentation, when other students are presenting, you can actually, start jotting down on the peer post assessment, right? So by the end of that day, you actually should, by when we close the lab session, you should theoretically have a completed post-assessment as well. So that way you are not wasting your time, because you are going to get busier as you get close to April 29th with, the practicals, other exams and all of that. So um, I strongly encourage you to complete the post-assessment as you listen to other student posters. Um, yeah, let me actually take one more look at over here. Yeah, so this particular link you're looking at, the poster PowerPoint link, is where you upload your final poster, right? Because when I grade it, I'm going to look at the notes I take while you are presenting, and I'm also going to take a look at and then subsequently, before finalizing your grade, I will look at your final presentation. So upload your PowerPoint presentation over here, right? Now I am aware that some of you are actually maintaining Google presentations, right? So if you just provide the link over there, right, I may not have access to that particular link, right? Because Google will, make you add me directly as a like, you have to share that, with me. Not just uploading the link might not properly work unless you clear access protocols separately. So rather than just pasting a link, download your Google presentation. If you're working on Google, download that Google version onto your hard drive as a PowerPoint and upload that as a PowerPoint presentation. The template we provided is not as a Google presentation, it's as a Microsoft presentation. So make sure you are actually uploading a PowerPoint file, a single slide PowerPoint file, not a Google link, right? Most often students forget to actually, share it, rather they paste the link. So the link does not work. Make sure you do that properly. Any questions? Any follow-ups? Okay, good. Let's go through this presentation, right? Yeah. A little bit over top. Right. So we are going to talk about today about gymnosperms, which includes non-flowering seed plants. So if you are looking at the phylogenetic tree, remember this is the same phylogenetic tree that we are using over and over and over. Make sure we have the chat window. All right, so we do know that these are the gymnosperms. Again, gymnosperms, that's not a single lineage, right? It starts over here, right? But we are going to cover several separate lineages of gymnosperms, right? But the interesting fact is all of those gymnosperm lineages, we are looking at 1, 2, 3, 4 over here, all 4 of those lineages share one recent common ancestor over here, right? Or rather over here, right? So all gymnosperms have a recent common ancestor, right? And the gymnosperm, all those gymnosperms are actually, a sister taxa to angiosperms, right? So since all gymnosperms have a recent common ancestor, right, they are monophyletic. Okay, all right. So and remember, right, they are also vascular plants, right? And if you trace back, the vascular plants evolved over here. So they are also vascular plants. But the advantage is now they actually have seeds. The characteristics that you're going to learn today that are that evolve in gymnosperms or in seed plants is actually presence of pollen. We see the first sign of heterophory, heterospory, and we will talk about that. You know what homospory is. Seeds, presence of actual seeds. Ovules, it's a new organ that you are going to learn. And the gametophytes becoming extremely reduced in terms of their duration in the life cycle as well as their physical size, right? The origin of seed plants can be dated to about 305 million years ago. All right. Moving on, again, this is a very similar phylogenetic tree. The purpose of showing that is for you to understand that seed plants are also vascular plants. Look at this particular category over here, and they are also part of the land plant lineage. They are part of the vascular plant lineage, right? And there are two groups of seed plants, flowering plants and angiosperms, and then the gymnosperms that are non-flowering plants. And that all of those seed plants evolution can be traced back to about 305 million years ago, which is also quite interesting that there was a gap about 100 million years between the evolution of vascular plants and the evolution of the actual seed. Moving on. Now, remember, guys, we are going to learn about a lot of new terms. So if you get confused, if you want me to explain something back, please do not hesitate to holler me, interrupt me, right? And make me explain back again. I do have the chat window open, but chances are I may not, see it on a timely manner. So hauling is actually the best way to go. Right. So gymnosperms, what kind of phyla are we going to learn? I'm circling the phyla right over here. These are the groups we are going to learn about. Oh, this number of species known, this is for FYI, for your information. We are not going to, unless I specifically mention the number of species found, I'm not going to test you on the number of species, right? But for a couple of occasions, I will mention, because, like in certain cases, we only have one species, one member, right? Unless I mention this again, don't memorize the number of species for each phyla. Some helpful hints to studying and making your lab notebook. Make sure to label everything, right? You can, particularly with respect to ploidity, right? Especially, like I kept mentioning, studying life cycle is the best way to understand these animals, these plants. So do label everything in your life cycle, right? And define the parts of the part of the words to make sure that you understand them easily. I did do this etiology last time, so keep doing that. Remember that plants produce spores via meiosis and sperms eggs via mitosis, right? So last time most of you mentioned that you're familiar with meiosis and mitosis. I'm glad that you guys are, because most students confuse that. And looking at your homework, only like one or two students got that wrong. So do recall that no matter what life cycle you are studying, what phylum you are studying, spores can be only produced by a sporophyte, right, through meiosis, right, where the genome number is halved from 2n to n, right? Sperm generation or egg generation or gamete production can only happen through mitosis through gametophytes, right? And mitosis is actually creating clones, right? It's kind of it does not change the genome number or the genome composition, right? It actually duplicates what you already have. Meiosis only occurs in reproductive cells, right? I mean, that's a universal statement, even for animals, right? Meiosis only occurs in reproductive cells, in reproductive organs, right? Only in the process of generating gametes, eggs and sperms. Mitosis can occur anywhere, right? Can it occur in reproductive cells? Of course it does. Can it happen in reproductive tissues, reproductive organs? Yes. And also in every non-reproductive cell, it can happen as well, right? Let me actually stop this automatic advancements. Right. Yeah. Yeah. So somatic is actually any cell, any tissue on your body that is not involved with sexual reproduction. Now, remember, there are vegetative tissues, right, that are helpful in reproduction. When I say reproductive tissues earlier, I'm referring to tissues that are performing sexual reproduction, right? Some somatic tissues like plant roots, rhizomes, and parts of leaves can contribute in vegetative or asexual reproduction, right? So that's when I say reproductive. In this case, I'm referring to sexual reproductive cells and tissues. Again, I'm not going to go over this. I did go over this part last time. This is again a revised etiology, right? Where I tell, a particular part of a name of the terminology and what it means, right? So remember to update your terminology, and this part will help you to actually, create a good terminology list for your lab notebook, as well as while you are preparing for your practicals. Well, definitely, even this might even help for your lecture exams as well. So gymnosperms is the group we are going to learn today. Gymnos mean open, naked, unprotected. That's what gymnos mean, right? Sperm, funny enough, refers to seeds, right? So not actual sperms, right? That term here, if we are referring to seeds. So gymnosperm refers to naked, open, unprotected seeds, right? Meaning, what do you mean by naked? Meaning it's not protected by, okay, it's not encased, it's not protected within another structure, right? It's open. Um, so gymnosperms, if they don't actually have, any casing to protect the seed, they should be housed, kept somewhere else, right? So they actually have cones, right? That actually keep those seeds, right? And the cone, scientifically, we call them strobilus. Remember, the term strobilus was introduced to you when we talk about certain ferns, right? Fernalize, right? Uh, uh, club club mosses, for example, a fernalide. And the structure, like the basic structure, is exactly the same. Now, in these cases, for the second time in the plant groups, you see sporophyte being dominant in the life cycle, right? Gametophyte now has become extremely reduced in terms of size, so reduced it's microscopic. Literally, you have to put the thing underneath a microscope to see what it is, right? And the sporophyte is actually the one you see when you say, oh, a pine tree, you're actually looking at the pine sporophyte, okay? And it is macroscopic. It's extremely dominant. Within the life cycle, it actually, for the 90% and above, you see the sporophyte as the most dominant stage in the life cycle. Gametophyte could be extremely small in terms of its presence, very short. Um, and all phyla of gymnosperm lineage are heterosporous, producing different spores, right? That is leading to production of male and female gametophytes. And we will talk about that with respect to examples later on, right? So remember, right now, in gymnosperms, you will not see homospory, right? And you will always see separate male and female gametophytes, separate male and female spores, right? Microspores and megaspores, right? We will talk about that later on in detail. These are the first plants with woody tissue. Evolutionary speaking, in plant evolution, right? These are the first plants with woody tissue. Now, woody tissue, what do you mean by woody tissue? Woody tissue are tissue that actually support, literally physically support the upright growth of the plant. Well, now remember, you did observe upright growth when we talked about ferns. Some upright growth among certain mosses, but they were short, right? They were not growing that tall. True, some of the ancient extinct ferns actually had much more taller upright growth under a completely different climate condition, right? Under very completely different physical and biological conditions, they did have taller upright growth. But none of those none of those ferns are actually reaching the same height you observe in gymnosperms, right? And in fact, it is, if you wanted to make a guess on the tallest plants that are living today, those are actually gymnosperms, right? Redwoods, for example. And we will talk about that, as we talk about redwoods later on. So the upright growth is actually created by a secondary growth of the vascular tissue. And we will show that in an example with more illustrations later on, right? Now, remember, angiosperms, flowering plants, also had woody tissue, but gymnosperms evolved before angiosperms. And then the last part is actually, this is there too, continue to have true tissues. Now, compared to ferns who also had true tissues, these guys actually have more complex, complicated, highly differentiated tissues. Well, one of those true tissues are needles, are leaves, and those leaves are actually either needle-like, like a pine needle, or scale-like, right? And they do not have flowers, right? In literature, in some of the photographs you are going to see, you might see the title flowers. They are not actually true flowers. You have to put quotations, right, to make because actual gymnosperms do not have flowers. They actually, if someone calls, oh, that's a pine flower, you are looking at the pine cone, not the actual flower. There are no gymnosperm flowers. Leaves do have a very thick, prominently thick waxy cuticle that help minimize water loss. And also, the waxy covering are slippery. So snow, when lands on the pine, it actually slithers off easily, right? So the problem with snow accumulation, it's not just snow itself. What happens is when it warms, if the snow melts, it will become ice, and the ice will freeze around the leaf blade. That might damage the leaf blade. And snow itself, if it accumulates for a long time over the pine needle, that will also prevent photosynthesis because it will cut off the sunlight supply. So it's important for the snow to slide off the needles, and the needle shape itself will prevent accumulation of the leaf, accumulation of stuff, snow or ice on the leaf blade, right? And generally, the cone shape itself will also help. The cone shape, the coniferous shape in most gymnosperms will also help to get rid of, to prevent snow accumulation. And also, if the snow accumulates, it will actually, add weight on the branches, and the branches will start breaking off. So I did mention that the gymnosperms have woody secondary growth, right? Secondary growth also happens not immediately when the seedlings arise from the seed, right? That's actually when the seedling first resurfaces from the soil. That's actually the primary growth. The secondary growth happens in older plants, right? Now, that secondary woody growth is present in both flowering plants and non-flowering plants. But in gymnosperms, in non-flowering plants, the woody tissue, the secondary supportive tissue, is softer compared to angiosperms. But that does not mean that the woody tissue in gymnosperms is less useful, right? There are some lumber industry and paper and pulp industry that actually has evolved around harvesting woody tissue from gymnosperms. Um, most, not all, most gymnosperms are evergreen, meaning they do not shed the leaves every season. So because of that, gymnosperms are also perennial, right? Meaning, unlike some of the mosses or some ferns, gymnosperms will actually remain above ground, right, across multiple years without any need to re-sprout from below ground every season, right? So those are interesting characteristics or remarkable characteristics of gymnosperms. Now we will look into different phyla of gymnosperms. First, ginkgo phyta. In general, we call them ginkgos or maidenhair tree. Maidenhair tree is actually the only non-member of that particular phylum, right? Now, ginkgos, um, they actually have separate male female trees. So that's actually meaning they are dioecious, right? They actually have male trees and female trees. And female, so if they have male female trees, you are going to have male female cones. Let me make sure I'm not missing the cycads. Nope, cycads are coming up next. Continuing on, so female trees will produce female cones, and they actually have fleshy scales that produce butyric acids, making them have a very pungent and putrid odor in the fall. So during the fall, when they actually are producing seeds, with the cone, they will produce the butyric acid. So because of the acid, you actually get the smell in the air, right? If there are several trees planted, you will smell them further away. And ginkgos, they produce pollen, and the pollination happens with the wind, right? And I said there's only one species, and that is actually this particular species, native to China, right? Endemic to China, right? And have very high medicinal values, actually true scientifically validated medicinal value, multiple of them. And they produce sperms, and the sperms are motile, right? And the motile sperms are also found in cycads, which is another group I will talk later on, right? Um, so this is actually, how their leaves look like close up, right? And they do undergo color change, right? And they are ancient plants, right? So there are several interesting fossil records, right, uh, showing the entire preserved leaf right over here. Right, let me take my pointer. Right, entire leaf, and also multiple leaves over here, right? So they are very ancient plants. They have been around for long. So I did refer to over here the male and female cones, and these are actually photograph of the male and female cones. They are quite different, right, morphologically. And this is actually the leaf, right? The leaves are not different between female and male plants, but the cones are the one that helps you to distinguish between male and female plants. Start thinking about what is the evolutionary advantage of having different male and female plants, right? And we will talk about that towards the end. I will pose a question, so that we can discuss what are the advantages, or if any, what are the disadvantages. Now, another phylum, Cycadophyta, that includes cycads, right? And by the way, cycads are actually something you might be familiar with because it's actually heavily used for home gardening and also for landscaping. It's also among the most ancient of the gymnosperm phyla, and most gymnosperms use wind pollination, just like the previous group, the ginkgos, um, and except cycads, right? So generally gymnosperms are wind pollinated. So the only exception that are not using wind pollination are this group, right? The cycadophytes that utilize insect luring to assist their pollination. Now, again, just like the ginkgos, cycads too have separate sexes, right? So there are male plants, female plants. So dioecious, if you break the term, oasis meaning a home, di means two homes, male and female living in two different homes or two different plant bodies, have a specialized symbiosis with a cyanobacteria in their roots. So these plants, for any plant, absorbing nitrates, right, is a big challenge. They are very rare and a critical nourishment. In order for them to enhance their ability to absorb nitrates, what they actually have is developed a symbiotic relationship in their roots with a cyanobacteria named nostoc. So the nostoc has specialized cells that can, now cyanobacteria is unicellular, right? And so these guys, nostoc, they produce colonies, and some of the cells in their colony are specialized for nitrogen fixation. They absorb atmospheric nitrogen, gaseous nitrogen, and convert that into inorganic nitrates. And those inorganic nitrates can be directly utilized by the plant, right? So nostoc literally lives inside the cycad root, the root of the cycas. In the image, you are looking at one of the examples, right? And you can see the leaves grow like palm trees. You might mistake this in a home garden for an example for a hybrid palm. It's not a palm at all. It's a cycad. And kind of in the center, in the leaf whorl, you will see the actual cone, right? And they do see, you could see them in the wild, right? I mean, they are not just a hybrid. They are pretty looking. So you might think, oh, they must be just home gardens, home garden plants. They are not, right? So in the wild, you could see them growing in clusters, right? Or in individual plants, right? The difference is actually, if you're looking at a horticultural variety, they are a little bit more prettier. Look at, the unstructured nature of those plants in the wild and see how structured and how pretty they look, right? And they actually are relatively shorter in the home garden varieties. And so they morphologically they resemble ferns with respect to their the leaf morphology, because the leaves blade divides into this pinnae, right? They have the palm-like appearance, so they might actually resemble fern in terms of the leaf morphology, or palms in terms of the overall tree structure. The trunk is actually barrel shaped. They are thicker, right? Even if the trunks are shorter, the trunk itself is actually very thick, right? And the entire trunk is covered with the leaf bases. So when the leaves get old, the leaf dies off, but the base remains on the trunk. The weird look you see in this trunk, right, is because of the persistent leaf base that does not completely shed off um, from the trunk when the leaf dies. The leaves are very tough, right? They're very like physically tough, have a thick hypodermis, right? The kind of the skin of the leaf is called the hypodermis, right? It's like a thicker skin, literally speaking. Um, and the stomata, they do have stomata, but the stomata are not superficial. They're actually sunken, right? And I will show you some slides to show what that means. The distribution is tropical and subtropical, right? And you do see them, they actually can grow very well in subtemperate climates, particularly not very well around here, but grows very well around Florida, right? Or in the southern part of the US. Now, interestingly enough, many gymnosperms have characteristic chemical compounds that they produce, right? So cycadophytes produce glycosides, right, which is actually a neurotoxin, right, meaning it could interfere with the communication among different neurons, right? Um, and the reason for producing a neurotoxin is actually to create a defense against pathogenic bacteria and fungi, right? And also against herbivores, right? Because bacteria and fungi does not have a neurotoxin, right? But interestingly enough, still those neurotoxins actually have massive major impacts against pathogenic fungi and bacteria, as well as against other um, animals, other herbivores that actually have neurons, right? Growing is extremely slow, right? I mean, if you wondered, right, how young those shorter plants are, they're very old, right? They just grow very, very slowly. Moving on to another phylum, Netophyta, G is silent, Netophyta, right? They were considered once the closest living relative to the flowering plants. But chances are that is actually not supported through molecular phylogenetics, right? Like among all gymnosperm, they were considered to be the closest living relative for the flowering plants for many reasons, right? One reason is actually the venation and the distribution of the veins in the leaf blade, as well as the cone being similar to actual flowers. But those are not really the closest relative to the flowering plants. We still don't know what it is, right? So they actually have two types of xylem tissues, right? Just like the flowering plants do, the two types of xylem cells are actually the primary and the secondary growth, right? That actually, the secondary is the one that produces um, the woody tissue growth, right? So that was another reason why people considered netophytes to be the closest relative of the flowering plants. One of the other reasons, right? And in fact, all of those, are woody plants, trees, shrubs, and vines. So they actually have different morphologies. They do produce, separate plants for male and female sexes, right? The best example for that is Welwitschia. The bottom picture you see over here, right? That doesn't look like a tree is actually a tree, right? It actually looks like a vine, it acts like a vine, but it is actually a tree. Now, other than Welwitschia, right, other netophytes can be either dioecious or monoecious, right? Meaning like some netophytes will produce both male and female cones on the same plant, and some of them will actually have separate plants with male and female cones. Another fun fact particularly about Welwitschia is that the leaves, right? What happens is actually in Welwitschia, you might think, oh, they have a lot of leaves. No, they actually only produce 2 leaves. What happens is the leaves actually break along the axis on the length of the leaf blade, and that split will continue to grow independently, and then they will split again and then keep splitting like that, continuously along the length axis. So ultimately, in a single plant, you will see the plant having multiple slender long leaves. They are not. They're actually just 2 leaves that kept repeatedly splitting, right, and continues to grow, right? So male and female cones, they actually are not flowers. This is actually a misnomer. I intentionally mentioned that so that they are not actually flowers, right? Those are male and female cones. Uh, um, and they are morphologically quite different, right? So you may ask me, okay, wait, if those are actually actual trees, not, spreading vines, where is the stem? The stem is extremely short, like few inches, embedded in the middle of this leaf roll, right? So you cannot, unless you actually sort of shuffle through the leaf blades, you will not see the actual stem. Continuing on to other example for netophytes, right? I'm showing you 2 genera here. And there are lots of, the reason why I'm not mentioning the English names is actually there are so many English names, right? It's too confusing. So that's why I am only mentioning the scientific names over here. The first example of double fertilization through the second egg fertilize, although the second egg can get fertilized, it dies off. So what I'm mentioning over here is actually that in the female cone, right, in a given ovule, you will get 2 eggs in netophytes, right? 2 well-developed eggs, and both of them can get fertilized, but one of them will die and only the other will continue to grow into an embryo, right? It will not produce 2 embryos, but 2 eggs can get fertilized in a given ovule. So the venation, if you look at particularly at a netum, right, look at the venation of the leaf blade. Look at how there's a midrib and there are lateral veins coming out of the midrib. Now that kind of morphology will make you think that is a flowering plant, but it is not, right? So that's actually make it look like an angiosperm. And another character that made us think that netophytes are actually the closest relative of flowering plants, but that's not the case. Phylogenetically, evolutionary, that's not the case, not supported by molecular evidence. Recent molecular studies indicate that similarities of this group to angiosperms is an example of parallel evolution, right? So where evolutionary close lineages evolve similar characteristics when they occur in similar environments, when they are under similar environmental stresses, they show similar characteristics, similar traits. But that does not necessarily show that that particular trait evolved from a particular common ancestor, right? So we know now for sure that netophytes are unlikely to be angiosperm ancestors, right? So the second example, ephedra, right? They produce a particular chemical compound called ephedrine, which is a chemical precursor to pseudoephedrine, right? Which is actually now that particular compound. There are 2 compounds, right? That and methamphetamine. So methamphetamine, you know that what is in the common English we called it meth, which is a psychedelic compound, extremely powerful addictive psychedelic compound. And then the first compound, pseudoephedrine. Now that compound is actually has two important medicinal properties. One of them is actually it's a decongestion, right? It can actually, if you have a nasal congestion with snot and all of that, this will actually help you to clear your nasal passage. The second importance is actually related to the decongestion. It will actually increase your alertness, sort of a caffeine like property, right? Which actually increases your wakefulness, alertness. So meaning that also that, so these compounds, these two compounds have sort of neurological effects as well as other potential medical effects as well. Continuing on to other phyla, Coniferophyta. Well, this group might be the one that you are most familiar with. Some of you might be familiar with cycads, particularly in home garden settings, but this one undeniably is the form of gymnosperms that you guys are aware of. Most of the gymnosperm species are actually in this particular phylum, right? I cannot tell you the exact proportion. I don't remember. I don't, that's not an important part, but more, a huge proportion of gymnosperm species are conifers. Some plants have both male and female cones on the same tree. So most of the conifers actually produce male and female cones on the same tree, monoecious. Few of them are actually dioecious. So all the reproductive structures are located inside this particular cone, right? And the images of the cone, the cone shape and cone size are quite different. I mean, I'm providing some photographs over here, right? So they can be that much variable. If you cross from the US to Canada, right? If you cross into the boreal biome, right? Conifers are the dominant vegetation you see, right? And if you keep moving into more northernmost boreal forests, it becomes the only woody plant you see, right? And so they're well adapted to extremely cold, frigid climates, right? And there are two kind of frigid climates, right, on the planet. One is actually northern latitudes, polar biomes, right? The second one is actually mountain tops, like high altitude mountain tops. In both environments, the climate is extremely cold. So these guys, coniferous vegetation, are well adapted to that kind of a climate. Extremely good carbon sinks. They are very good at very efficient at photosynthesis because they can photosynthesize year round. Well adapted to winter, remain active during winter, don't become dormant. And I did mention that they are evergreens. Well, there are few conifers that do shed leaves partially or fully, but most of them do not. Most economically important group of gymnosperm because for extracting medical important compounds for pulp and paper industry, and to produce resins, so in timber industry, right, which is largely for extraction of lumber and timber, like the secondary product that you can gather from conifers is actually this extracting the chemical compounds like resins, gins, and other other medically important compounds. So this picture actually sort of is depicting the four groups of living plants that we have covered so far. And notice how the only group with the dominant gametophytes are the mosses, right? And the gametophyte gets progressively smaller as the plants evolve, and the sporophyte in most evolved plants become more dominant and more larger, right? And also once the the sporophyte initially in the evolutionary history were dependent on the gametophyte. But now you notice that the sporophyte is completely independent, right? And now in terms of seed plants, you also notice that the gametophyte is now dependent on the sporophyte. Remember, in ferns, both gametophyte and the sporophyte were independent, right? They can photosynthesize on their own. But now in seed plants, gametophyte is dependent on the sporophyte. Okay, so the rest of the figure you can actually study by yourself. I wanted to reflect back on these characteristics. These are synapomorphic features, derived traits for all seed plants, both angiosperms and gymnosperms. Reduced gametophytes, small sized gametophytes, microscopically small gametophytes, both male and female gametophytes are microscopically small. And they are protected by the sporophyte and they are nourished by the sporophyte, right? We will talk about where exactly those are found when we talk about the life cycle. Strict heterotrophy, heterospory, right? Microspore is the one that is representative of the male spore, giving rise to male gametophyte. And literally, microspore or the male spores are smaller. Megaspore are larger, much larger, and they will produce the female gametophyte. Ovules, a new organ that you haven't seen in previous plant groups, right? It's structurally looks like this, and it actually has three components, right? The outermost covering, the protective covering, integument. Integument is a term you should be also familiar with. We use the same term for animals, and it is diploid. Um, and then inside that you see megasporangium is a term you are already familiar with respect to other plant groups. And then inside the megasporangium, you actually have the megaspore, right? This the one of the heterosporic forms that we just discussed. Pollen, right? So that's actually so now you are learning where these spores are located. Where is the megaspore? The female spore is located inside the ovule, right? And do notice that the megaspore is haploid, but the megasporangium is diploid. Pollen, right, is the pollen grains make water unnecessary for fertilization because for the pollen grains to spread, they don't require water, unlike in the sperm, you saw in ferns and mosses, right? So that is a really important character because now gymnosperms are independent of access to water, right? Liquid water, standing water for fertilization to take place. Now, where is, microspore? That's actually microspore is actually located inside the pollen grain. Seeds, well, when seeds survive better than unprotected spores. Spores have the ability to survive harsh, severe environments, but seed can do it better for a longer period of time. Certain seeds can go dormant for years, couple of seasons in certain cases, but years in many cases, right? And in fact, you can store seeds for many years, and then they will still germinate if the adequate conditions are met. And seeds can be transported longer distances, which will help that particular species to expand their distribution range, right? The structure of a seed, you have a seed coat, like the basic layout of the seed is very comparable to ovule because it is actually the ovule that transforms into a seed upon being fertilized by a pollen, right? So inside the seed, you have the seed coating outermost, and then inside you have a huge food supply, and then innermost is the developing embryo. Again, these 5 characteristic features are unique to all seed plants. We did mention several times of the stem, the woodiness. Right now, let's talk about what that means. Again, first group of plants to produce the secondary wood, which is actually coming out of the secondary xylem, right? Allows the plants to grow much taller. So because of this, the secondary xylem is actually, it's able to provide a better strength to the plant, to the stem, so the stem becomes stronger and can grow taller, right, without snapping into two. So the job of the woody tissue is supportive. Now, in order to produce secondary xylem, right, a wood, you need to have another meristematic tissue, that produces that particular secondary wood. So vascular cambium is a meristematic tissue, right, that is ring-like in arrangement, right? If you look at this particular cross section, right, the outermost part is the bark, right, and then the vascular cambium is actually this very thin line of cells, right, that I'm showing with my pointer, that is circular in arrangement. That ring of tissue is responsible for producing xylem cells towards the inner part of the stem and phloem cells to the outer part of the stem, right, towards the outer edge, right? And then if you further look into the structure of the cross section of a gymnosperm stem, you actually see the wood has two components, right? The zap wood and the heartwood, right? The pit is the centermost part, which is actually very not very strong tissue. It actually can deteriorate quite fast, and create sort of an empty hull, right? The heartwood is actually the most stronger part that you have this secondary tissue. Sapwood is actually much more weaker, right, in terms of its structure. So the vascular cambium is actually a secondary meristem, right? What is a meristem? Meristem is a set of undifferentiated cells that makes up a tissue that can, the only job or its only ability is to undergo cell division through mitosis, right? Repeatedly undergo mitosis. Those cells will remain young forever, right? They will never lose the ability to undergo mitosis, right? They will keep doing that over and over and over, right? Those cells are undifferentiated. Because of because the cells are undifferentiated, they can through cell division produce more cells that later on will become differentiated and specialized into a particular function. In the woody stem, right, there are live cells and dead cells. Phloem cells are live, right? The vascular cambium, the meristematic tissue itself, is live, right? And then the cork cambium, it's another meristematic cell that can produce the cork, right? Those are the only living tissues in the woody stem. Everything else are actually not alive, they are dead. Now, during spring, right, when wood is produced, right, that wood spring in a stem tends to be larger, right? They produce more, larger xylem cells, right, with a larger cavity. The summer wood is actually the opposite. They tend to be smaller, right, thinner. So because the spring wood is larger, thicker, and the summer wood is smaller and thinner, right, you can clearly see in a cross section the spring wood part and the summer wood part. That will help you to actually count the development of rings, annual rings on a cross section of a tree. And that annual ring count helps you to determine and assess the exact precise age of the tree, right? So like if you count the number of, what is easy to count is the number of spring wood, because those are larger and thicker. The number of spring wood is actually going to be analogous to the number of the years the plant have lived, right? Or if you just count the summer wood, the thinner, smaller rings, you can again enumerate the age of that particular tree. Now, can you do the same in a tropical climate? You cannot, because in a tropical climate, there are no spring, no summer, right? So you cannot separate out annual ring growth. So it's very hard to do the same thing for a tropical tree, but for a temperate tree, because of the seasonal differentiation, because of the seasons are different, how trees grow during spring and summer are different, right? So because of that, you can clearly count those rings easily, which you cannot do in the tropical climate. So like I just mentioned, if you look at over here, spring wood, right, actually has these cells that I am actually pointing with my laser pointer are the xylems produced during spring season, right? They are larger cells with a bigger opening, right? If you look at way up over here, you can see they actually have smaller cells, smaller cavities, and more thicker cell walls, right? So summer wood is going to be thinner and smaller. Now, Tracheids are the type of the xylem cells you find in most gymnosperms, right? The name of this xylem cells, specific name of this open, xylem cells, or this, thick walled xylem cells, all of them are called tracheids, right? And most of the tracheids in a xylem are actually not living, right? They as immediately as they are produced with the cell wall thickening, they die. The reason is actually the job of the xylem is to transport water and minerals. If you actually have living tissues in the tracheids, that will resist water movement. So that's why it's extremely important for the tracheids to be the dead tissue, right? So that it will not impede the movement of water and minerals. Another part is, what are those large canals, those circular cross sections you see? Those are called racine canals. Racine canals in gymnosperms carry a sticky, thick substance that are composed of two things: one, rosin, and turpentine. Rosin is a solid which can get dissolved, right, when a particular solvent is present. The solvent that actually is that solubilize rosin is turpentine, right? So turpentine is the solvent, rosin is the solid, right? What is the point of these things? Okay, first of all, racine canals are literally pipes, right? They are ducts. And in some images you will, or in some literature, you will see them named as racine ducts. The same thing. They are not cells like tracheids. They are actually um, lined by multiple epithelial cells and creating a continuous duct that carries resin, that delivers rosin and turpentine together. We call that resin, throughout the plant body. Do you find them in stems? Yes. Do you find them in branches? Yes. Do you find them in plant leaves? Yes. Right. And what happens is, if the plant get damaged, right, the resin canals gets exposed, so the resin oozes out with rosin and turpentine together. Turpentine is volatile, right? It will vaporize, leaving rosin behind, and that will solidify and sort of heal the wound of the plant body. Do not mix up the rosin with the pine sap, right? Pine sap is actually what you find in the vascular tissues, right? Resin canal is not the vascular tissue. Vascular tissue is a combination of xylem and phloem together. So the actual rosin canals, the resin canals are elongated, tube-shaped intercellular spaces, intercellular ducts that are covered with epithelial cells, right? So the resin canal themselves are not cells. Vascular rays, these things that you see are called vascular rays, right? Or just rays. Those extends in a stem cross section. They extend radially, like a radius, right? From the center, from the pit towards the bark. They radiate across the stem. Their job, their live tissues, their job is assisting transmission of contents of the vascular bundle laterally, right? So the vascular tissues, xylem and phloem, their transportation is more vertical, right? For lateral transportation, what happens is these vascular rays will actually conduct those components laterally. Couple of things about the secondary growth. Secondary growth allows plants to produce woody tissue and to grow thicker. So because of the secondary growth, stems will enlarge in size, roots will enlarge in size, they become thicker. Most of the secondary growth are xylem cells that will become dead as they mature, like I just mentioned, right? So secondary growth mostly produces xylem, some phloem, but the biggest job of the secondary growth is to produce more xylem that will subsequently die as they mature. Actually, like quite immediately, they will die. In the late winter or spring, there is a lot of availability of water and nutrients. So the xylem cells produce this time of year are large. What does that mean? Like, remember the spring wood I mentioned, right? During late winter to throughout the spring, what happens is the snow is melting, the resource availability is increasing, right? Plenty of sunlight, plenty of nutrient because the leaf decomposition has released nutrients, right? Snow melt has released water. Some groundwater is oozing out, so there's a lot of surface water available. Soil moisture is rich in water, so there's a plenty of resources available. As a result, the xylem cells produced during spring and winter time are going to be larger because there's a lot of resources to conduct, and the plant requires a lot of resources that should be transported to the roots, to the stem, to the branches, to the leaves. So as a result, the xylems are now actually going to be larger in size. That's why the springwood is actually larger. Then what happens during summer is actually water becomes scarce, nutrient becomes limiting, so there's little resources available, right? And the plant now is actually well grown. It actually, has, collected the critical compounds. So there's sort of a less need, less demand as well. So because of the resource limitation, the xylem produced during fall and later summer are going to be smaller in size. Okay. I did show you this image already, and I did mention the difference between sapwood and the hardwood already. So the hardwood is actually much more stronger part, right? Sapwood is actually the relatively weaker part, and the hardwood is actually brighter in color, darker in color compared to the sapwood. Now, sapwood contains xylem cells that are actually conducting water and minerals, right? They are dead, but helps to conduct mineral and water. Hardwood, the center part, now sapwood is the peripheral part, hardwood is more the central part. Xylem cells that are losing their function and becoming filled with tannins and minerals. What happens is actually xylem, even will they're dead tissues, right? They will transport water and minerals. What happens is with time, those minerals that it transports, other dissolved material that it transports, will become solidified, will become suspended and precipitate within the xylem, right? And sort of block it, clog it. So those older xylem tissues will not be able to transport minerals and water because they are blocked, right? But blocking becomes an advantage because that kind of blocking with tannins, with resins, and with other organic and mineral compounds will add strength to the xylem tissue, and that strength will also help to to provide more rigidity, strength to the stem itself. The springwood, also known as early wood, are the xylem cells that are produced in spring, and the springwood, late wood are actually the xylem cells produced later in the summer, and you already know which one is thicker, which one is larger. Annual rings is actually the collection of the springwood and the summerwood that happens within a particular year. Pith is actually the centermost collection of cells, which is actually part of the original primary growth that happens when the plant emerges from the seed as a seedling, and that will eventually become what, as the plant continues to grow, as the plant accumulates more secondary growth, the pith becomes crushed towards the center, right? The ray, I just showed those, radiating cellular parts. Those are the vascular rays, cells that can bring materials horizontally through the stem instead of vertically, right? Vertical distribution is handled by the vascular bundles. Those are vascular tissues. Rays are the ones that are collecting stuff from the vascular tissues and transporting them elsewhere horizontally. Vascular cambium, I also mentioned what that is. Those are the cells responsible for, those are meristematic cells responsible for producing phloem to the periphery of the stem and the root, and xylem to the inside of the stem and the root. Bark is collective, it's multiple tissues collectively that that you get from the vascular cambium towards the outer edge of the tree, right? Which actually is a part that is literally peeling off when you see an older tree in the nature. Cork is the outer bark of the tree, right? And then cork cambium is a cell layer. It's a meristematic cell layer responsible for producing the outer cork. Why is it important? Because the cork or the bark part keeps peeling off. You need to replace that peeling tissue, right? That's why cork cambium is important, to keep continuously producing cork to account for the peeling bark, right? So we described this particular part already, and you can clearly see in this one that the springwood is the xylem are larger, right? And that spring wood itself is thicker compared to the summer wood, right? Resin and resin duct, I talked about that already, right? It's sort of repeating the same structure, same components over here. Any questions regarding the basic introduction, guys? Any questions? Okay, no. So now let's actually moving on to the more interactive part, right? So for this particular part, I do want your active participation, please. So please do turn on your cameras. I would appreciate it. So let's actually move on with this part now, right? And I would really appreciate if you could actually turn on your camera so we can actually have a more active conversation, right? Thank you. So let's go through this part, and I'm going to ask you several questions as we go. And please do ask questions because I do realize that there's a lot of new terms that I'm throwing at you. Okie dokie. The first exercise is actually the key. Guys, Matt and Emily, give me a nod. Should I do one example, or are you much more comfortable with knocking this off on your own now? One example. Thank you, Emily. Let's take a look at the first cone, right? Look at the description. Cone is dry, like, I mean, physically it's dry. The length, it's longer than 3.5 centimeters, and the scales are papery in texture. They're actually thin and papery, like if you feel about that. Okay, let's go. 1A. Corn fleshy or look berry-like, right? Like raspberry-like. B, corn dry. What is it? Go to 2 or go to 3?
Go to 3.
Very good. Go to 3. Cones up to 3 centimeters in length or bigger than 3 longer than 3 centimeters.
B.
Correct. It is definitely B. So wait. I did it wrong. No, I did it right. So go to 7. On the 7, scales thick and woody. Scales thinner and somewhat papery. What are we going with that one?
7B.
7B. We are looking at scales thinner and somewhat papery, right? And we go to 8, and scales thin, papery, and lacking brackets. Scales thin and subtended by a 3-point bracket, right? If you look at over here, you don't see any subtending, right? There's no evidence of that happening, right? So we're actually going by the first point, larynx, right? So that's actually basically how you go about. Let me see if there's anything I want to point out. The description is on the side. So grape-like is actually this kind of appearance. And if we ask you about something about something that actually has a, no, I think this actually is self-explanatory. Everything else is quite self-explanatory.
What is a 3-pointed bracket?
Yeah, I'm actually trying to look for a photograph over here. 3-pointed bracket is actually, when you're looking at this particular part, like this is a bracket, right? At the edge of the edge of the bracket, you will see 3 spines, right? If you look at over here, right, you actually see these barb-like things, right? Sorry, this spine-like things, protruding, like 3 points coming off the edge of the bracket. Okay, that's what that means. Any other questions regarding that?
But the hint says no barbs.
Yeah, no barbs. The barb is actually, those are not bulbs, those are actually sort of spines. That's kind of emerging from it. A bulb is actually a much shorter spine. There was one. If you look over here, can you see right over here on the top of this particular, let me get my pointer. Can you see what's going on over here? That is a very conspicuous pointy bulb, right? That is what you call bulb. And over here we are saying, right, cone is dry, more than 3.5, scales are stiff and barbed, right? And that is what a barb means. And in other ones that you can see here, you can clearly see the barb. Each and every one of them has the barb. This is a huge barb. You can clearly see that, right? It's not 3-pointed bracket, right? When I say pointed, you clearly see pointed, right? You know, the long arrow-like points, right? When I say barb, you are looking at a shorter spine, right? Any other questions regarding any of those verbiage or anything? Okie dokie. And this is the terminology that I'm going to cover for the rest of the exercise. Now to the life cycle, right? So life cycle is a little complicated. So I'm going to go over the overall, the bigger picture first, and then I will actually cover two of the more critical, smaller parts within the life cycle separately, right? So we are looking at over here, this is actually your mature sporophyte, which is actually what you call the pine tree. You are going to look at the male cones and the female cones, right? The female cone, you know, are going to contain this very unique structure called ovule, right? And so if you look at the cross section of this female cone, right, you are going to look something like that, right? It's going to have these brackets, right? And you know, there are many names: brackets, megasporophile, right? And you know, on the base of the bracket or the megasporophile, you are going to see this ovule, right? And if you blow up one of these structures right over here, it's going to look like that, right? So remember the components of the ovule, right? Ovule has the megasporangium, right? And inside the megasporangium, you are going to get the megasporocyte. Remember, megasporocyte is a new word. Megasporangium isn't. Megasporocyte is a new word. It's actually 2N, right? So it's diploid, right? And the megasporangium is also 2N. Now remember, all of those are located on the sporophyte inside the cone. That's part of the sporophyte tissue. So that's why they all are diploid, right? And then looking at the male cone, you know, male cones, by the way, are smaller, right? Male cone looks like this in a cross section, right? And you know, inside of the male, if I take one part of this microsporophile, right, and look at that, it actually has, we are looking at a microsporangium, the equivalent of the megasporangium, microsporangium. Inside the microsporangium, you are going to have a lots of microsporocytes. That's another new word. And note that all of those are diploid. Again, part of the sporophyte. Makes sense why they are diploid. Now, the job, what's going to happen to those sporocytes, right? Those sporocytes are diploid, right? If they were to produce spores, what must happen is undergo meiosis. Makes sense. You are looking at a sporophyte. Sporophytes undergo meiosis to produce spores, right? So when first, what happens to megasporocyte? What happens when it undergoes meiosis? It produces a megaspore or a female spore, right? Actually, how many? They initially produce 4 megaspores. 3 of them die, 1 survives. Microsporocytes, there are so many microsporocytes inside the microsporangium. There's only 1 megasporocyte inside the megasporangium, right? So many microsporocytes inside the microsporangium. That's a contrast, right? And then when microsporocyte undergo meiosis, they produce pollen grains, right? And inside the pollen grain, you actually have the actual microspore, right? So What is the difference between a pollen grain and a microspore? I will get to that point later on, but you will notice that in the literature, they are used very interchangeably. Now, what you see is those pollen grains are extremely small. They're taken up by the wind, pollinated by the wind, and some of them will get deposited onto the ovule, right? The ovule, on the top of the ovule, there is a tiny opening called micropyle, and I will show that later on. And it will land on the megasporangium, right? And then the megaspore, the surviving megaspore, meanwhile, will undergo some development. That will produce the megaspore. The job of the female spore is to produce a female gametophyte, right? Also known as mega gametophyte, right? And that mega gametophyte will produce 2 archegonia, right? And with each archegonia with a single egg. And then meanwhile, the pollen that landed on the megasporangium will produce actually a pollen tube, and that pollen tube will insert a sperm onto one of the archegonia, right? And immediately the other archegonia perishes, other egg perishes, right? And subsequently fertilization will happen. Now remember, at this particular step, pollen grain is haploid. Megaspore is haploid. Now fusion will make the zygote diploid, and zygote will develop into an embryo. And then the ovule undergo transformation and making the now the ovule will become a seed, and the seed will grow into a seedling, and the seedling growth maturation will continue for many years, producing a mature sporophyte. That's the completion of the life cycle. Okay, now I actually simplified it. There are certain details that I will go on what's happening with the pollen grain and what's happening in the megasporangium. Those are the parts that is actually also very important to know. Please do stop me and ask questions when things are not clear. So the female cone, right? A single sporophyte will contain many female cones. A single female cone will contain many ovules, right? This is actually how a female cone looks like. In a diagram, before fertilization, it looks like this, right? It actually has this central axis, and then there are these brackets, right? And then on the top of those brackets, right, you will actually have an ovule, right? And these brackets are already they undergo different names, like sometimes we call them scales, we call them brackets, right? But yeah, know those two names. And the job of those brackets, right, are actually to house the ovule, right? And you can see clearly over here, right, that ovule is naked, it's open, it's not housed inside a flower, right? That's why we don't consider these cones as true flowers. They are naked, they are open, right? Now, if I, put an ovule, a young ovule, into a microscope, it looks like this, right? So a pine ovule, right, shown in this picture, actually has a megasporangium, also known as nucellus. I do want you to know the terms that I mentioned because it appears in literature in different ways. It's painful. I don't know why people use different terms, but they do it anyways. And it is the same as the megasporangium. Now I'm going to give you a warning. How many of you have, give me a shout? How many of you have covered pine reproduction in your lecture? Okay, one of you is raising your hand, so at least one of you do. Now, Emily, tell me, or others who actually might have covered this, did your professor mention that nucellus is a remnant form of megasporangium? Anyone remember?
I don't remember.
So, either way, your lecture professor might mention that. You might read that in textbooks, you might read that in literature. I am not sure whether I'm convinced whether that is correct. I'm worried about using the word remnant, right? It doesn't, make sense to me. So I'm going to tell you that you will read that word, but I'm little skeptic about using the word remnant. Okay. Now, moving on, um, the other parts of the ovule are the external most part is this part is the integument, right? It's multi-layered, right? That is the integument, which subsequently will produce a seed coat after fertilization takes place. Then you have this megaspore mother cell, right? Also known as megasporocyte, right? And then that megasporocyte will subsequently undergo meiosis and produce 4 daughter megaspores, right? Or female spores, right? And then now don't get confused. The pollen grain you see over here is not part of the ovule. What happened over here is a pollen grain is over here because some pollen have landed on the ovule and actually successfully entered the ovule, right? It's not literally or biologically part of the pine ovule. Now, what we show over here is actually, it takes a very long time, the minimum of 2 years, sometimes more, for a female strobilus, a young female corn to mature, right? They will grow in larger in size, right? Initially, you will see these brackets or scales are overlapping with each other, right? And then what you will see is when the ovules are more mature, right, the scales will actually literally open up, right? And when it's fully mature, it will fully open up, right? And when after the fertilization, it will open up even more. So it takes time. I mean, look at this time scale, right? From this point to this point, it'll take about the minimum of 2 years, right? And by end of 2 years, it's ready to release its seeds, right? What happens if they don't get fertilized? They'll drop the corn. That's it. Now, it's important to understand very carefully this part of the life cycle, right? Starting from this particular picture, what you're looking at is a mature but unfertilized ovule ready to accept pollen grains, right? So you get the integument, right? Again, think about where this is coming from. This is the female corn, still sticking onto the female gametophyte, female sporophyte, right? And then you have the, what you see is actually the megasporocyte that was inside the megasporangium have undergone meiosis and produced 4 megaspores, and 3 of them have undergone degeneration, right? And the one of them remains, and the rest are absorbed back to the megasporangium. Okay, and then that megaspore, what happens is it will, as the maturity continues, the female gametophyte, also known as the megagametophyte, right? Um, uh, the megaspore will mature into a female gametophyte or a megagametophyte, right? So see how small that is? Notice that the megagametophyte now is literally inside the female corn, dependent on the sporophyte, microscopically small, right? You will never observe it in the external environment, unlike what you saw in terms of ferns and mosses, right? And that single female gametophyte will produce 2 archegonia. I don't know why they produce 2 archegonia, because subsequently only one of them will become fertilized, right? The one that does not get fertilized becomes degenerative, right? So, and now what happens, like while the megaspore undergo that development, right? So the megaspore produce the female gametophyte, the female gametophyte produce 2 archegonia, each archegonia will produce egg, right? And that egg production in archegonia is through mitosis, because remember, it has already undergo meiosis to produce the megaspore, right? So the megaspore can only undergo mitosis, right, for the subsequent developments. Now, that's the female part. What happens to the male part, right? The male cone has produced pollen, right? And the pollen grain enters the ovule through a micropyle. This opening in the integument is called a micropyle, simply a hole. That's what it is. And then what happens is this pollen grain will create a tube, a literal tube that will develop and grow into the megasporangium of the ovule, right? And that will actually and then of course that developing, growing pollen grain, right, will actually have two cells in it. One is the tube cell that is responsible for producing the tube. The second one is the generative cell, which is responsible for producing the sperm, right? So in fact, the generative cell will produce two sperms, right? Now, it's really interesting. There are two sperms, there are two eggs, one in each archegonia, but only a single ovule will only support a single event of fertilization. That is really strange, and I'm not sure why it happens. Could be an evolutionary stasis, a carryover, but let's not worry about that. The pollen tube grows into the developing female gametophyte. Yes, I just mentioned that. And the developing pollen grain can now be termed as the male gametophyte, right, or microgametophyte. So, and a sperm is discharged into the archegonium, right, that contain in the egg, and randomly out of the two archegonium, or randomly out of the two eggs in each archegonium, one of them will get fertilized. Immediately, the other one will be aborted, right? So now, once fertilized, the zygote grows into an embryo. Zygote is unicellular, it's diploid, it becomes an embryo, which is multicellular. And the female gametophyte tissue, right now, remember what happens? This is the megasporangium. And here you are looking at the developing female gametophyte. The developing female gametophyte over here will take up the space that was previously occupied by megasporangium. So the female gametophyte tissue will take over and fill up the megasporangium, right? And that female gametophyte, why it is happening? Why it is the importance of development of the female gametophyte upon fertilization? Because that will become the nutritive tissue, the food reserve. Starch, it will be filled with starch. In certain cases with protein, in certain cases with lipids. So that will provide nourishment for the developing embryo, right? And the embryo continues to grow, will keep growing, and that will gradually become embedded and ingrown into the female gametophyte. Still, where is this happening? Inside the ovule, right? As this happens, the like unfertilized ovule became fertilized ovule when fertilization happened. And after fertilization, as the zygote becomes an embryo, as the female gametophyte becomes larger, the fertilized ovule becomes a seed, right? Now, while that happens, let's see what happened to the integument. Integument hardens, becomes thicker, and becomes a seed coat. Its job is to protect the developing embryo. And of course, the whatever the remnant of the female gametophyte will become nothing but the food supply, right? Now remember again, all of this happening inside the female cone, right? None of this can be observed in the naked eye, right? Unless you like take out a cone, remove a bracket, and then take out the ovule and then observe it under a microscope, you will not see any of this happening. Now, also think about the fertilization part here, right? About the male cone and what's happening in the pollen, right? So the pine pollen grain, right, has a tube cell and the generative cell. Generative cell will produce sperms which can travel, right? And through what? Travel through the pollen tube, right? And the pollen tube will create a channel to ensure proper migration of the sperm into the female gametophyte. Now, this developing, growing, maturing pollen grain can be considered the male gametophyte, right? So it's actually it's a very simple structure, very small structure, you know, just like the female gametophyte, right? So it's simply the job of the male gametophyte is to create sperms and make sure the sperm is properly delivered into the female gametophyte. So again, the female and the male cones are quite different. Female cones are larger than the male cones, right? And they both occur in clusters, right? And if you look under a microscope, it's also really interesting. You can clearly see that this male, inside the male cone, right, there are lots of microsporangia, right, and lots of microspores inside them, right? And then and then you can see the sizes of the equivalent female structures are quite different as well. Now this is not a part of this comparison, is not part of your lab, but I just mentioned that, I included that just for your knowledge, for your comparisons. You look at the different parts of this pollen grain or developing male gametophyte. It actually has these two big, huge air-filled structures. They are called air bladders, you know, like a urinary bladder. Instead of storing urine, they are storing air. The point of that is actually to ensure long distance transportation of pollen to ensure cross-fertilization, right? And inside this middle structure, what you get is actually the tube cell containing the tube nucleus and the generative cell containing the generative nucleus, right? And those are found in this inside structure. Do not want you to worry about this. Come on. This prothalial cell, I do not want you to worry about that at all. That's kind of on the sort of the bottom of that developing pollen grain. Don't worry about that at all. A few more things about the male cone and the pollen. Most gymnosperms are wind pollinated. You know, they release the pollen they produce to the wind, which means they have to produce tons of pollen. If you wondered in the previous image why there are so many microspores or microsporocytes inside those male cones, the reason is actually you need to produce so many pollen, right? And wind pollination is not very accurate. It's not a given. It's not guarantees. It does not guarantee fertilization. Now, animal pollination guarantees fertilization, higher probability of fertilization, right? But if you compare that with wind pollination, wind pollination has a very low probability of successful pollinization. That's why you need to produce so many pollen as a cone, as a male cone. The pine pollen grain in this picture is showing both the tube, right? This is the actual tube, right? And inside that you get the tube nucleus, right? And then and that tube is produced by the tube cell, right? And then you see over here, right, the generative nucleus and the generative cell that is responsible for producing the actual sperms, right? And then on either side you get the air bladder, right? That helps to suspend the pollen in air. Remember, male cones are always smaller in size than the female cones. The reason is it's not that expensive to produce pollen, doesn't take a lot of nutrient to produce pollen, so there's no need to make it larger. And also the second problem is if your male cone is larger, it will impede pollen release, right? So those are the two reasons why those are structural differences. Here, the next part of the exercise is for you to actually take a look at each individual part that I just went through, right? So now, interactive component, guys, work with me, label these points with me. What do you call this entire young plant that you locate inside the embryo, or rather inside the pine seed?
It's the embryo.
Yeah, that is correct. That is the embryo, right? And then what do you call these parts that are actually responsible for photosynthesis? Randolph,
Um,
give it a shot, my man.
what's the start? What's the first letter?
They're the first leaves. Okay, they are the first leaves. And
Is it
eh,
the pith?
sorry.
That's not. Oh no, I was gonna say the pith.
Oh, no, 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 no. Remember, the pith is actually, so you are right now looking at the pine seed.
Okay.
Pith is actually the center of the stem, right? And the center of the root, but we are not making you study the root system. So these are the first leaves. Now, can someone tell me the specific name we give the first leaves that actually emerges out? They are called the cotyledons, but I don't want you to remember that term. I don't think it's even in your lab manual, but just saying them the first leaves is good enough. Nutritive tissue, this entire part, all of these parts, what becomes the nutritive tissue? Can someone tell me?
What part are we talking? Like the inside or?
Yep. Look at what I'm looking at, pointing with my pointer.
Okay, would that be like the mad gamma fights?
That is correct. That is actually mega gametophyte or the female gametophyte, right? Remember, female gametophyte became the nutritive tissue, right? Oh, and well, the rest of the parts are much more easier. The last part, the outer covering have been removed from the seed, right? This does not actually have the outer covering of the seed because you will not see the inside if you had the seed intact. And the name of the outer covering of the seed, it's very simple.
The seed coat.
Correct, seed coat, right? So, yep, that's correct. Seriously? Okay, now let's label this with me again. Now, little help over here to you, right? On this side islet image will help you to figure out what these things are. And you, like you just mentioned, right? What should this nutritive tissue that I'm pointing should be? Go on.
The gametophyte.
Female, be very precise. Okay, female gametophyte or mega gametophyte, right? And yes, I mean, these are going to be the photosynthetic leaves, and yeah, the rest of the structures are quite self-explanatory. Moving on. So answer me this question, right? So this is the embryo, right? The embryonic cells are quite small, right? You can see tiny dots all over the place. And in contrast, the gametophytic tissues are much larger. Why? It's completely okay to make a mistake when I ask these questions. You will learn from that mistake, trust me. Why are they differently sized? Is it related to their function?
They have to store a lot of food.
That's correct, right? The mega gametophyte or female gametophyte job is to store food. So where are they going to store food? You have to store the food inside a cell. You should not store the food like in the empty spaces or the extracellular spaces. That's a problem, right? That if you do so, they will actually undergo hydrolysis automatically. So in order to, if you want to store more food, you need to have larger cells. That's the reason. Well, why embryonic cells are smaller? Well, what the job of the embryonic cells are actually to remain undifferentiated and is responsible for the primary growth, right? They will keep dividing and produce new tissues, and the new tissues will undergo differentiation. So early embryonic cells will always be smaller because their job is not to differentiate but to keep reproducing. So whatever the cytoplasm they produce, whatever the extra protoplasm they produce, right, they will be always be redistributed through cell division, right, into daughter cells. So they will continue to be smaller. All right. Further, a few more labeling. So here we are looking at a mature male strobilus or mature male cone cross section, right? So now just to help you out, I'm not going to go over this because I have a image over here for you to label, right? So the questions are, what are this overall structure is called, and the things you find inside this that are diploid or that are haploid, what are those are called, right? So remember, if I say these are diploid, that's a different name. If they are haploid, that's a different name, right? And then this particular structure that actually sort of holds this thing, right? What are their names? I mean, I'm not going to go over it because the answer is right over here, right? But I do want you to understand those names and be able to label these independently. Now, here we are looking at a female cone, specifically aiming at a single ovule, right? So this particular cell, the cell that goes through meiosis to create 4 daughter cells, and 3 of those daughter cells die, 1 remains alive, right? What is this cell is called?
I have
So
a question about the last slide.
yeah, of course, ask.
Are the leaves where the containers sit, are those the same between males and females, the name?
That's a great question, right? That's a great question. So we are looking at they have different names because they look different, right? So the leaf-like thing is called a fill, right? And if it is male, it's micro, right? So it is going to be microsporophyll, right? So yes, there are differences, right? Yeah, micro because it's male, sporo because it's actually producing male spores, fill because it's a leaf, microsporophyll. Any other questions on this one? We are all set. Okay, back to this question. What do you call this particular cell that will undergo meiosis?
Megaspore.
Megaspore is the cell that is produced after meiosis. So before meiosis?
Megagametocyte.
It is not. Sorry, say it one more time for me. You said megagametocyte?
Yeah, yeah.
Well, it is megasporocyte. Right, megasporocyte, right? Because think about it, it is producing megaspores like you earlier said, right? So megaspores should come from megasporocyte, right? So and then the container that holds all of this, what's the name for that?
Megasporangium.
Very good, right? I mean, it depends on how I call that. Since I'm only pointing to this particular part, Emily's answer is right. That is megasporangium, right? And then and then this particular part, the outer covering, multicellular outer covering of the megasporangium is going to be, now this is before fertilization. So, you know, there are 2 different names, after fertilization, before fertilization. What do you call that? Starts with letter I.
Integument.
Correct, integument, right? Now, last question. What this entire structure is called what? Start with O.
Ovule.
Yep, correct, ovule, correct, right? So this is actually naming the same thing, right? So over here, right, this is the ovule, like earlier I asked that question already. Leaf upon which, or the bracket upon which female reproductive structures sit, right? Now that is going to be some fill, and it is a sporophyll, and it's female structure. So it is megasporophyll, right? So female and male cones are going to have very, very different sizes. I mean, we just did an exercise number 1 where we used the cone shape to identify different species, right? Let's take a look at the male gametes, right? The pollen, and try to understand them. So this is actually a microscopic slide of different, a lot of pollen grain, and each pollen grain in different developmental stages. Some of them are very young, like this one, right? Now, these structures that on either side of the actual pollen grain are known as what? They are filled with air. They're called air bladders. Okay, those are called air bladders, right? And then and the name of this entire structure, right? I need you guys to answer, right? The name of this entire structure is
Pollen grain.
correct. That is pollen grain. The cell that is responsible for creating sperms. There are only 2 cells inside, right? And this is what? This particular cell inside the pollen grain. Starts with letter G.
Gametophyte.
Now, gametophyte is not a cell. Remember that, right? That's an actual multicellular structure. Generative cell, right? And look at this, the name of this particular outgrowth that comes out of the pollen grain that goes into the female gametophyte. What do you call this particular structure? Starts with letter T.
Pollen tube.
Yeah, it's tube, correct. It is tube. You can call it pollen tube, right? And the specific cell that produces the pollen tube is what? That is easy. It is producing a tube. It's a cell, tube cell, right? It's very easy, guys, actually. If you get the logic behind it, like everything else is so easy to remember. And this mature pollen grain, can I give it another name that is actually growing into a female gametophyte? Can I give it another name other than mature pollen grain?
Would that be considered a zygote?
No, it has not. Zygote is only when fertilization takes place, right? So this is actually, we don't, this is actually not, we are not even looking into the zygote, because zygote is not observed on the pollen grain. Zygote is only kept inside the ovule. So what would you, it has sperms, right? It's producing sperms. So what kind of a thing produces sperms? Male something, right? So male sperm is a gamete, right? A tree producing gamete. What do you call that?
Microspore.
Microspore was located inside this, right? Before all of this happened, right? It is actually male gametophyte. This entire structure can be called male gametophyte or mature pollen grain. Right. We already did a very similar labeling over here. So only one thing I wanted to point it out over here is actually, can you see, right? You know, so this here we can see the female gametophyte is growing, right, and becoming the nutritive tissue, right? That actually replaces something else, and that something is now restricted to a very small part of the entire ovule. What tissue is this?
Sporangium.
Very good, right? What specific sporangium? Megasporangium, right? That is megasporangium, right? Okay. Now we are looking into actually the plant leaf blade, right? And in fact, I will actually show you the cross section step by step over here. That's pine needles. If I were to take a cross section, like cutting across the leaf needle like that, I'm going to see something like this. This is a diagrammatic view. In real life, it's going to be looking less pretty like this, right? So first thing I want to show you is look at these canals that you find all around the entire leaf margin. Those are the sunken stomata. The canal-like thing is actually like a pit, right? It's a pit. So the stomata are located on the bottom of that pit. The reason is actually that will prevent water loss. That's the advantage of having stomata on the bottom of a pit, like the sunken stomata. The purpose of the sunken stomata is to prevent dehydration. And then you have the epidermis as the outer covering, right? All over the leaf blade, except where you see the stomata, right? And then the tissues inside the stomata that sort of fills up the rest of the plant in the needle interior are actually sclerenchyma, like the ones that are immediately surrounding are called sclerenchyma. Again, that name I do not expect you to know, right? All the cells that you find, right, you can call them mesophile. Mesophile are any cell that fills up the space, right? That's a universally acceptable name for any plant. Now, this particular very unique bundle that you see in the center of the pine needle are actually the part that contains the vascular bundles. You can see two vascular bundles over here. One vascular bundle, another vascular bundle, right? And so this thick cell walls that you see over here that are larger in cell interiors with a very thick cell wall, right? Those are called, actually if I zoom that part, a single vascular bundle over here, it looks like this, right? This part, you can see they actually are stained in pink. They have very thick cell walls and a very large interior. Those are xylem, right? See, xylem is found even within leaf blades. Other side is actually phloem. You can see those cells are smaller and the walls are thinner, right? Last part is actually, what are these? Look at these canals with multiple layers of epithelial cells surrounding them, right? Like this, like this, like all over. Those are resin canals or resin ducts, right? So resin ducts are not just limited to stems. You find them in needles as well, right? So now based on that, I want yourself to label this diagram on your own, right? Now let's look at these different primary and secondary growths, right? So in a very young 1-year-old pine, right? You don't have the secondary growth, you only have the primary growth, right? So you actually see towards the center, right? You have the pith over here, right? And then you see this, thick-walled, large opening cells that are the xylem, right? And then towards the exterior, you see much more like a smaller layer of cells. Those are actually the phloem, right? And then out of exterior to phloem, you always see resin canals. Now, the fact that you see xylem innermost and phloem outermost, that is universal, right? No matter what plant you are looking at, for any vascular plant, that would be the arrangement, okay? For angiosperms, that would be the arrangement. And then you will see these resin canals, right? out of to the next to phloems, you will see these large resin canals, and then you will see this outermost tissue that's actually called the cork, right? And underlying the cork is the cork cambium, and cork cambium is a meristem tissue that produces the cork. And then if you blow up a section of this plant cross section over here, right, that's what you're looking at, you will see the same structures, right? Resin ducts all over with different sizes. Thin-walled vascular tissue are going to be the phloem. Thick-walled large vascular tissue are going to be xylem. And those very large cells of the xylem are actually tracheids, right? So based on that knowledge, right, I want you to be able to, sorry, this is actually, I'm not going over it. This shows the same structures, right? And then remember, I mentioned that bark is a collection of tissues. So that bark has the cork, and then the cork cambium immediately under the cork, right, which is a live tissue. Phloem next, vascular cambium next, right? And then this resin duct and the pit interior most, and then interestingly, xylem you actually see in multiple layers. Remember why? Because xylem is produced in every year, right? A thin layer in the summer, a thick layer in the spring. That's why you actually see xylem layers as multiple layers, right? But phloem only has a single layer. The phloem is produced in subsequent growth, but there are no differences in the phloem thickness. Phloem is always produced in the same size, same thickness, same cell morphology, right? So that's why you don't see multiple layers of phloem, right? And only fewer phloem cells are produced, but more xylem is produced. So in a mature pine tree cross section or a stem cross section, you will see multiple layers of xylem, and each layer is a single annual growth. But you don't see the same regarding phloem. Okay, now based on that knowledge, I want you to be able to label this particular diagram yourself. Now here again, you can do your own, to best of your ability, keep counting the growth rings, right? And that number of growth rings corresponds to the age of that particular tree. Now this is actually a question for you. Last two slides are, display questions. You can see this is a really cool image of pollen, right, being emitted by a male cone, right? Most conifers are wind pollinated. Discuss one advantage and a disadvantage of wind as the agent of pollen dispersal. Right, I want to hear from you guys, right? Give me an advantage. What is the benefit of wind pollination?
Long distance travel.
Excellent. It actually carries a lot longer than an animal could, lot longer than even water could, right? There are some plants that are water dispersed. Good. Another advantage or a disadvantage?
You could take it to higher locations to reach the female cone.
That is correct. It will take there's less guarantee, less probability for each pollen to land on a female gametophyte or land on an ovule. Yeah, that probability, that chance is lower. That's what you said, Randolph, right?
I'm saying like some cones are, that's even higher up. They can take them to the higher. The wind would take them up there.
That's actually correct. That is true as well. The relative locations for male and female cones are also different. That's actually an excellent point. Yeah, and correct. Anything else? Any other advantage or a disadvantage?
There's some potential for self-pollination.
That is also correct. So the height difference might actually, but sometimes it will prevent it, but it will also facilitate it. But that is true. If it is wind pollination, right? Wind can blow in any direction. So still you could undergo self-pollination. That is true, right? At the same time, it could land, on a completely different, like anywhere, right? There is no direction of guaranteed delivery of the pollen. So yeah, self-pollination could happen when wind is the only mechanism. Correct. So those are good explanations. So the last question here is actually, before we go to the homework part, all of the species in this phyla, Gymnosperm phyla, have plants that are either male or female, right? Discuss one reproductive advantage and of having male and female reproductive organs on separate plants. So dioecious versus monoecious, right? We are mentioning that lots of gymnosperms are dioecious. So tell me one advantage and one disadvantage of having male versus female plants. Now, I'll give you a hint. What Emily just mentioned can be brought back here. Any idea?
There's no risk of self-pollination?
Correct. Exactly. Right? Because the same plant does not produce male and female cones. So that's actually a huge advantage. It actually increases the genetic diversity. Anything else? Any advantage or a disadvantage you may want to think here?
Would a disadvantage be that it would be harder for them to reproduce?
Yeah, because you have to find another plant, right? And if it is wind pollinated, you have to find another plant of the opposite sex. Yeah, the probability or the chance of fertilization happening is now lower. That's not incorrect. That's a good point. Yeah, those are two good points. Yep, that's basically all about that particular activity. The next thing is actually to go over the homework. Any questions before we go to the homework? Alrighty, good. Let's channel through the homework. So here what we are asking for you is answer the questions below using information from today's lab. Some of course, you are working individually. Refer to the integrity policy of your lab syllabus for more information about the consequences of copying. Generally, like, I don't find this problem, particularly you guys are actually, now working individually than when you are in person. Some questions require use of your textbook. Yes, only when information is not in your textbook or lab manual, use legitimate online sources. To be honest with you, since I cover a lot of background information and spend so much time on the exercise, you probably won't have to go outside, but if you do have to, do that by all the means. Complete the table using your notes, blah blah blah. So scientific names of species observed, right? Starting from charophytes, right? Charophyte examples are given because we didn't particularly study charophytes, right? So mosses, ferns, and pines, right? We went for many examples. So pick the example that I detailed the most and mention the scientific names. Remember, when you are mentioning the scientific names, they should be italicized since you're typing them in. And then the phylum, yeah, it's going to be, Bryophyta for mosses, Pteridophyta for through ferns, and then pines is going to be conifers, right? So here it's easy if you list true ferns because I have talked a lot about true ferns, less about fern allies, right? Like club moss or stuff like that, probably avoid them over here. Go by true ferns. And then body, well, what do you expect over here, right? So the body of a moss is largely going to be gametophore or gametophyte, right? Which contains phylids, rhizoids, blah, 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 right? And the ferns, well, it's actually, the body is going to contain the sporophyte, right? And actually the sporophyte contains megaphiles or leaves, right? and then megaphiles in the case of the strobila, but otherwise true leaves, right? And of course, the true roots and sometimes stem and the rhizoids. Pines, well, the structures that is actually responsible for reproduction are going to be leaves, right? Now remember, like it says structures where photosynthesis is happening, not just the body, right? So forget about what I mentioned regarding the stem or the rhizoids. They are not contributing to photosynthesis. It should be the parts that are actually contributing to the photosynthesis. For mosses, that are the phyllids, right? And then for the ferns, that is going to be the leaves and megaphiles to a certain extent. And for pines, it's just the leaves. I do know there are certain ferns where the stem is photosynthetic, but go by the main feature over here. Anchoring structures used for attachment. Mosses only had a rhizoid. Ferns had roots and the horizontal stem, which is actually the rhizome, and the pines just the roots. Looking at structures for gaseous exchange, remember mosses had spores on the gametophytes and stomata in sporophyte only. Ferns and pines all had stomata and just stomata. Just an additional thing I want to mention with respect to pine. Now, additional thing, this is, don't confuse. There are sometimes stomata-like structures on the stem. They are called lentils, right? Those are openings, right, in the stem that helps gaseous exchange in the stem, but that's just an additional information for you. You might learn more about that in the lecture. Structures protecting from desiccation. Well, the cuticle can be applied to all of those. And actually, yeah, actually that should be good enough. But in pines, you actually have sunken stomata. And yeah, you can actually mention even the vascular tissues are actually, organized in such a way to prevent water loss. If you remember, the xylem of the vascular tissue has a thicker wall. The reason is to prevent water loss. Habit, the growth structure, the growth morphology, right? For the mosses, well, they are short, right? Grows in clumps. Now, don't remember the clump, the clump morphology, right? Ferns, they grow taller, right? Relatively taller. Upright structures. Now remember that they too make clumps. Pines, right? They are tallest of all, right? They do go upright of all, right? And pines don't grow in clumps. They grow in an individual trees, right? And over here, this part is actually, again, I'm not going to go over myself because I have several label diagrams where you can actually do this on your own, right? And the next part is as well, like you are looking at several examples. Here, this part, right? You are looking at all plant produces spores. However, sporangia that produce them are very different in 3 groups of plants that we examined so far. Compare the tree genera provided here representing 3 different groups by filling in the chart below. Where appropriate, use relative terms such as least, more, most, or small, small, smallest. So, moss example, fern example, and the pine example, right? So the sizes, right? Now, what you have to notice is actually, I'm not going to give you the answer, but I'm going to give you certain directions. As you ascend the evolutionary tree, if you go to more derived trees, more recently evolved trees, right, more evolutionary recent branches, the sporophyte size becomes smaller. Sorry, sporangium characteristics, size of the sporangium relative to the sporophyte, right? So the sporangium itself becomes smaller, right? When you move into much more recent lineages in the tree, right? And then sporangium characteristic, the number per sporophyte, how many sporangia are found per sporophyte. Now, that feature, right, the number of sporangia per sporophyte, is very fewer in younger very fewer in older evolutionary branches. As tree evolution continues, you actually get more ploidity. Now, ploidity, remember, right, in the sporangium is located on the sporophyte, can only be what? Regardless of the group. Is it haploid or diploid? Is sporangium?
Diploid.
Correct. Always diploid, no matter what example we are talking about, right? Located where? Located on sporophyll. Remember the sporophyll. Phyll is like a leaf, right? So sporangium occurring on the top of a leaf-like structure was only found in ferns and pines, right? And in the pines, actually, we even give them different names, right? In certain ferns, we give them different names. Not in the case of mosses, right? And then hetero versus homospory. Remember, we mentioned that heterospory is only found in what group? What is heterospory? Having two different sizes of male and female, micro versus mega. So what is the only group that show heterospory here?
Pines.
That's it. Just pines. Well, that's basically it. How gymnosperm disperse their pollen? I mean, we talked about the pollination part over here several relative times with respect to some of the display questions. So what I want you to do over here is discuss, like elaborate on those advantages and disadvantages, right? Yeah, I mean, the other ones actually are self-explanatory. I don't have to provide you any guidelines. It's more basic general knowledge, thinking about if it is wind dispersed, you need to have some structure that will assist the seed to be carried further away. So then that's why you actually get wings, right? Well, what is the problem if you're going to be wind pollinated? Well, you need to be lighter. Your seed has to be lighter. You cannot be excessively lighter. Well, because if you are excessively lighter, your embryo is smaller, so your survival is smaller, and of course your nuclear tissues are going to be also smaller, right? And your seed coat are also going to be thinner, right? So you will have a lower survival chances, less food available for the growth. So yeah, you will be dispersed over a longer distance if you are a smaller, lightweight seed, but you will compromise the nutrition supply, embryo survival, and the resistant to environmental stresses. But if you are actually putting a lot more nutrient, if you are permitting the embryo to grow larger, and if you are putting a thicker covering, then you are having a larger seed which will survive better, but it's not going to get carried away. One way to overcome the larger seed size is having additional structures that will suspend them in the air longer, that will help the seed to hover around in the atmosphere longer, right? The wing evolution is actually kind of an adaptive feature for larger seeds to be carried further away, right? But still you have a problem, right? Does it make sense for a smaller seed to have larger wings? Well, no, then the seed is small itself. It can be carried by wind its own. It does not require large wings. If you have large wings, it's going to add more weight, right? Wings are weight, right? At the same time, if you actually have a larger wing, larger seed, probably having larger wings is helpful because now you're adding a little bit more weight. Well, the thing is already heavier, but the wing structure will actually act like a helicopter. It may be able to carry it longer weight. It will hover for a longer distance. It will actually get suspended and remain in the air for a longer distance, right? There are cooler ways that happening in gymnosperm world, particularly in the part of the world that I'm familiar with. There are actually seeds that you can literally see flying in the air, right? So That is basically it. Any questions regarding the homework or anything we covered so far? Anything on presentations, exams? Okay.
There's a bonus question.
Yes, we do have a bonus question at the bottom, right? How is it possible that bird dispersed pine seeds don't get digested by the birds? The question is actually, I almost skipped it. I usually don't help towards the bonus question. Since Emily raised it, I will put some guidance. So any animal, there are so many animals that are actually dispersing seeds. I know that because I study how animals help dispersing seeds. The advantage is it's great because you can actually make the animals walk around. Animals actually will carry it to a particular place, probably to places where the seed germination is guaranteed. And sometimes seed, when the animal eat it, it is deposited in the environment back with poop. Poop is nourishment for the seed to grow, right? So because of that, there's a huge advantage when an animal is helping seed dispersal, right? And in this case, in terms of pine, right, generally, like not just for pine, for many seed plants, the seed dispersing animals don't digest the seed. They don't, right? They will eat the fruit, right, or the ovule, right, or the part of the corn, and will poop the seed intact. Think about how come the seed does not get digested. It makes sense to digest the seed. Remember what you find inside the seed: a growing embryo filled with nourishment. The female gametophyte filled with nourishment, right? Very critical nourishment like starch, oil, essential oil, and proteins. So makes sense for a particular bird or another animal to eat the damn thing, right? To crush the seed and eat the actual seed, crush it and eat it. But that does not happen most of the time. Most seed eaters in general, right, do not damage the seed. Only seed eaters that actually damage the seed are rodents, particularly non-native rats damage the seed. Most seed eaters do not. Birds largely do not damage the seed. So the question is, what kind of adaptations do you see in the corn, in the ovule, right, that will prevent seed damage? That's actually basically the question. And you can also think about, what does digestion require? Well, you need to literally break down the seed, right? On one hand. So you might want to see presence or absence of a particular physical digestion mechanism in the seed dispersing animal. Two, other than physically breaking apart, chemically breaking it up, right? Well, it requires specific enzymes. So if certain enzymes are absent, maybe the seed will not get digested. So think about those lines when you answer that question. Alrighty, that's actually basically what I wanted to explain. And we have 15 minutes more. Yeah, today it took some time because it's a lot of work to cover. If you do not have any questions, I can let you guys go. Oh, hey, James?
Oh, yeah.
Yeah, so I received the confirmation regarding your material.
Oh, yeah. They got it. Sorry.
Yeah, yeah. So yeah, just want to let you know before you scoot. So yeah.
I completely forgot to let you know. Sorry about that.
Yeah, no worries.
Thank
All
you.
right. Yeah, all right, guys. So if you don't have any questions,
I don't think I do.
yeah, scoot away.
Thank you.
Of course.
Thank you.
Thank you.
Thank you very, very much. Have a great day.
Yeah, of course, Jared.
Have a good one.
Hi, Professor. I was just gonna ask you for
Mhm.
since I have advising with you anyways, I was
You
just
do.
gonna hear You said between the 5th and the 8th and the 12th and the 15th, correct?
Yeah, like in my email, I gave you like, it's
Yeah.
like next 2 weeks
Yeah.
after April 5th, um, from Monday through Thursday. That's correct.
Yeah. So one of them has a file connected to, because I haven't picked out like any courses for next semester yet.
Mhm.
It says fall 2021 only electives.
Yeah.
Like, are those like the only things that we can choose to? I don't know.
No. So let me clarify what we mean by elective. Now you are going to take you are going to take ecology, right? So
Yeah.
ecology is not an elective; it's a required course, right?
Okay.
So that's not listed there because you guys we are required courses, core biology courses like bio 121, 122, ecology, cell biology. They will be always offered in that particular semester. So anything that is required, the 5 classes that are required, and you are BA or BS.
It's a bachelor's, so BS, I think.
I think you're BS. I'm pretty sure you're BS. So yeah, those courses, because for BS you have to take genetics, for BA you don't have to take genetics. So ecology is the one that you should be looking out for.
Okay.
Yeah. And the other courses you need to think about, Annie, is actually the non-biology courses. What are you doing right now for chemistry? Where are you?
142.
Oh, you're taking one. So you took 141 already,
Yeah.
and you are taking 142.
Mm-hmm.
Great. So you should be thinking about taking organic?
Yeah.
Yeah.
I haven't really looked into. I haven't like made a solid schedule yet for them, but I didn't get to take physics this semester, which I've wanted to, so I might have to work that into.
Let me tell you before physics, right? I'll come to the physics part. It's a good question that you raised. So keep organic 1 because general chemistry is completed and you are doing, I assume you are doing well. And so that is definitely something you should not miss, organic 1. How about math? Where are you in math?
Wait 1 second. I'm taking the second calculus course, I believe.
So with that, your math requirement is taken care of.
Yeah, 142.
Yeah, good. So physics 1 and 2 are not required. It is only required if you are trying to go to medical school, dental school, vet school, right?
Yep.
So if you are not planning to go to those schools, right? If you're planning to go to like PA school, nursing school, some programs require you take physics, some courses do not require. So depending on what school you are going to go to for any of those other health sciences, right, you may want to think about taking them. Now, let's say that you don't know right now, but after graduating, after doing a job for a couple of years, working as a scrub or whatnot, you decided, yeah, I want to go to nursing school, or yeah, I want to go to PA school. You can take physics 1, 1 and 2. I mean, what is your closest community college? Quincy, right? You can take there. You can take it there. So,
Yeah.
the point I'm going to tell you is, like rather than trying to take a course that you don't require. Now, do you plan to go to medical, dental, vet, any of that?
I think right now I'm currently like rethinking my plans. I'm really not sure anymore.
So if that is the case, you probably don't have to take those.
Yeah.
Right. And then again, let's say like right now you don't know, but in your final year, you say, Dr. S, I wanted to go to medical school. Well, then let's take physics 1 and 2 at BSU.
Yeah.
Right.
Yeah.
Right. Even after graduation, you can take 2 more courses. You can take courses up until like you can like delay your graduation, like you can work but delay your graduation until you complete like couple of courses. You can take physics after that. So but seems like where do you live, Annie? Do you live in Quincy?
No, it's Quinsigamond Community College. It's like out near Worcester.
Oh yeah, so you're a little far. Yeah, you're
Yeah.
about an hour away, right? Hour and a
Usually
half.
I live on campus though, so.
Oh, you do? Okay. I mean, it depends on like, I mean, if you like wanted to, like in a summer, go home and then take a close to home, you can go
Yeah.
to, there are options. So the point is, I advise you right now not to worry about taking physics since you are undecided what to do.
Okay, so focus on like the non-elective and then orgo and stuff like that.
Right. So the focus, let's go to, yeah, Ogu definitely yes. Math, you are done. Ecology, definitely yes. Ecology will be fun in person. Like last time it was virtual. That was
Yeah, I'm not a big fan of online
it's
classes.
fucking. You have no idea how much I hated when I was teaching. One of my favorite courses to teach, I taught it virtually. That's unbelievable.
I can notice like a big difference between what I actually like learn in person. Like my grades starting like freshman year and like the first half of second semester freshman year, and then like now it's just like I can't do this anymore.
Yeah, you lose the interest, don't you? Right?
Yes, I'm so done with it. I do not like it. I feel like I'm like struggling to learn anything. I need like a certain structure built around my classes, and I feel like I don't get that online. So
Yeah, I think the learning, like I mean we are science. You are a science student and I'm a scientist, right? We are the most hands-on people in this university, right? How can we teach something like that? See, like I mean, imagine any bringing a pot of fern to you and explaining these things and showing you a photograph. Like,
I
can
know
I?
that's what I was saying. I was just thinking when we were looking at like the seeds, and I was like, oh, this
Right.
would be so much easier to understand if we could actually like hold them and look at them.
Oh yeah, it's fun too. Like I mean, you will be more amazed when like when we do that. Like it's even